we'll start into chapter 6. Okay, so this chapter is called Tour of the Cell, and basically what we're going to do for this chapter is we're going to look at all the different components that make up the cell at the organelle level, right? So we talked about some of these organelles, and you guys did some labs on these, right? We did the mitochondria and the nucleus and the chloroplast and things like that. So we're going to talk about what the organelles are, what their function is, how we determine their function, and specifics about the larger parts of the cell. But before we begin, we're going to make sure that you guys know how we are able to study these organelles. And most of the reason that we're able to do this is because we can use something called a microscope. And what does a microscope do? It magnifies things, right? But it also magnifies them with a resolution that you can see that's coherent, right? Because a magnifying glass makes things bigger too. But what's the problem with that? When you get it to a certain size, it gets blurry, and that's no good. So, what kind of microscopes do we use in the lab? Compound. And so that means that there's two lenses. One is closest to your eye. What's that lens called? And the one that's closest to what you're going to see, the object, is called the objective lens. And those two work together, so the powers multiply together to give you your final magnification. So if the objective says 50, the ocular is 10, what's the magnification? 50 times 10 is 500, right? So you guys have been using the scopes in the lab, and you know that there's one that says 5. So is that 5x? The objective, the red one, says 5x on it. Is that 5 times? It's 5 times 10. That's 50 times magnification. Good. So let's talk a little bit about magnification and resolution. So you guys understand magnification means making things bigger. But just because you make it bigger doesn't mean that it's clear, right? You can't. Resolution is defined by your ability to distinguish between 2 points, right? So if I were to draw 2 dots on the board, like so, those are separate dots, right? But you walk further away, and eventually it's going to look like one dot. When it does, then you've lost your resolution. Does that make sense? So there are microscopes. If you go to Fry's Electronics, you'll see that they sell microscopes that go to 1200x. Are they lying? What's the maximum magnification that's on your microscope? 1,000, right? And that's because that's the maximum magnification that we can use. So the 1200x at Fry's Electronics, right? Are they lying to you? Do you think it's 1200x? It is 1200, but what's the problem with that? It gets blurry, right? So you lose resolution. So it's completely useless. The microscopes we use are the best optics that they have available for light microscopes. So what do you think the maximum magnification is? It's the ones that are on your scopes, which are 1000. Okay? So, and the reason is because, and we've talked in lab about wavelength, right? And so light is waves. Remember we talked about visible light in there? I just want to make sure that I did talk about this with my lab. Did you guys, for Mike's lab, did you talk about that too? Wavelengths of light? No? Well, you're not in his lab. Yeah, in my lab, yeah.
We did? Which lab?
We didn't? Alright, well, that's okay. Well, that's good to know. I'm glad that you told me that. So, okay, so we're gonna do this in lab. And one of the things that's important for you to realize is that there's four forces in nature, right? So there's gravity, and then we have the strong force, which holds the nuclei together, and then we have the weak force that holds atoms together. Those are like chemical bonds. And then we have the electromagnetic force. And the electromagnetic force that you're all familiar with, you use it every day, it's all based on waves. So the only difference between radio waves, right, that you use to listen to your radio, and microwaves that you use to cook your food are the length of the wave. So everybody's seen a wave, right? So wavelength is the distance of the crest to the crest of the wave. Or this is the same, the trough to the trough. That's the same distance, right? So this here shows you these are really long waves. So radio waves are really huge. They're mountainous waves, right? Really big. And these get shorter, so microwaves, what's a micro? That is an exact number for micro. 10 to the minus 6, which is 1/1,000,000th of a meter. So those are relatively small, right? And then we have visible light, which is right in the middle there. So we have infrared, which are bigger. The wavelengths are bigger than visible light, which is right here. And visible light lies between 700 and 400 nanometers. So what's a nano? So 10 to the minus 9, or a billionth of a meter. So everything that you see lies within 300 billionths of a meter of wavelength. That's pretty small. And then outside of that, things that are smaller, you can't see. That means ultraviolet light, right? You guys can't see ultraviolet, I assume. We'll test it in the lab. I'm pretty sure nobody can see that, or the federal government would probably be trying to find you. X-rays, can you guys see that? Walk around, check out people's bones. Hey, you got a tumor in your, no? And then gamma rays are so short that they're able to pass through the earth without touching any other atoms, nuclei, or electrons. All right, so that's the range. We have big mountainous waves, really tiny waves, and in between there is what you can see. Everybody got that? So when we use a light microscope, what do we depend on? If I turned all the lights off, could you see the specimen? So how are we able to see it? What do we use? We use light, right? And that means that light bounces through those lenses into your eye. That means that there's a wavelength that it has to take up. It takes up space. And so the reason that you can't make the magnification any greater is because you can't fit any more light in that length of waves. So if you want to increase the magnification, what can you do? You can't change the wavelength of light. It's always that distance. But what can you do? You want to fit more waves in there, what do you have to do? You have to use something that has a shorter wavelength, right? And so we can use electrons. Electrons have a smaller wavelength. So you probably heard of an electron microscope. That's why we use that, because it has a shorter wavelength than light. All right. So let's go back to everybody understand the difference between resolution and Magnification, right? I might ask you on the test, what limits the maximum resolution of the light microscope? What is it? Wavelengths of light, right? Visible light. So we can overcome that using the wavelengths of electrons. And so in this case, the resolution of an electron microscope is 0.2 nanometers. So here, it was 0.2 micrometers. So again, what's the difference? What's the difference between a nanometer and a micrometer? 1,000, right? So if this maximum is 1,000x, and we can go down 1,000 folds more, then what do you think the maximum magnification of an electron microscope would be? Right, 1,000 times 1,000 is 1,000,000. That's exactly right. So at that level, we can see things that are really small, right? We can see viruses. Probably seen the image of Ebola that was taken with the electron micrograph. But we can't get so much. You can see HIV. We can't get so much smaller than that, right? We can see big molecules, but not. You would never be able to see strands of DNA or lipid triglycerides or anything like that we've been playing with, right? But there are microscopes that can go down even further. I don't think your book talks about these, but these microscopes are called atomic force microscopes. Right, so there's a laser confocal microscope that probably can do about 1,500x, but the atomic force microscopes, they can see down really low. So the way that they work are basically, you guys, I know this is kind of old school, but everybody knows how a record works, like on a record player, some vinyl. So the needle moves up and down, right? And it resonates that, pitch or whatever on the thing and turns it into sound. So the same thing with the atomic force microscope, except the needle on that thing is one carbon atom wide at the end. So it's a diamond, right? And it's whittled down to one carbon atom at the end. So it gets drug over things and it moves up and down, and it can look at things like atoms, for example, because it's so small. ASU has these, but the buildings have to be built especially for those microscopes. It has to be in 50 feet of bedrock. It can't be, all the walls have to be insulated so that there's no electromagnetic interference, because anything that makes that thing move, right, even a distance of an atom would make the resolution cruddy, wouldn't it? So you got to take into account all this stuff, and those aren't cheap. They're millions of dollars each. The buildings have to be built around them. All right. So this is your book has kind of the reason that you need to know this is because it talks about the different things that you look at using a specific microscope. So let's say that you wanted to look at HIV, for example. Do you think that you could look at that under the light microscope? So what would you need to use? Electron microscope. Or let's say you wanted to look at the structure of a glucose molecule. What would you need to use then? Yeah. So the confocal scanning tunneling microscopes, they use lasers, so they can do about 1,500x, and they're kind of cool too because they can focus in different planes, so you could actually look at a living cell like it's being sectioned. That's why it's called tunneling, because it can focus on, let's say you have a cell, right, and it's like a sphere, like 3 dimensions, but it can focus on one plane of it, so it looks like it's sliced, a slice cell. Even though it's still alive, you can look through it as individual slices because it focuses on a different plane. Those are pretty expensive microscopes too. So anyway, all the way down to, say, frog eggs. You guys in your lab, we talked about some algae cells, right? Some algae eggs you can see with your naked eye. But below that, right, the inside of cells, the organelles, things like that, we can see with the light microscope, and you guys demonstrated that. But some of the small bacteria you can't see with the light microscope. Mitochondria, pretty close to the limit of resolution. So what did we do since we couldn't see the mitochondria? Yeah, well, we used that colorimetric, that color changing test. Did you guys in the other lab do that? All right. So DCPIP. And then below that, right, viruses, proteins. This is a little far-fetched. Like the book is a little ambitious, saying that you can see down to atoms with a electron microscope. That's not true at all. It's probably cutoff is right about here, and then below that you need something like an atomic force microscope. All right. So. Two different kinds of electron microscopes. One is called a transmission electron microscope, and the other one is called a scanning electron microscope. They work different ways. Basically, when you have a transmission electron microscope, let's say that you wanted to look at a tumor, like a cancer tumor. So the surgeon would remove that, and then a scientist or oncologist or lab person would take it and embed it in a piece of plastic called paraffin, right? And then you would take a machine called a microtome. You guys remember what that means? Micro, small, and what's tomey? To cut. So a microtome makes small cuts, paper actually like 1,000th thinner than a sheet of notebook paper, and it will slice those off of the cells, and then you can take it and put it in a machine, and it will coat it with gold or lead particles that the electrons bounce off of, and then you get images like this. So because it's sliced into thin sections, it looks like a two-dimensional image. This is actually the cells that line your small intestines. They're called endothelial cells. These are cilia, and when you eat your food, right, these cilia beat and they push your food through your body, and as they go along, it takes the nutrients that it can out. And you can see the inside of these cells. You guys see that?
So that's just a flat image of it.
Yeah, so this is a sliced image. So if you want to see the inside, what we would call an ultrastructure of the cell, you would use a transmission electron microscope, because that allows you to see inside, because you're slicing the cell. Does that make sense?
Did someone donate their body to go through something? I saw.
Wouldn't surprise me. Yeah, I haven't heard, but that wouldn't surprise me. I know that if you go to like those body exhibits, that it's probably not a microtome, but it's pretty thin sections where they have people that have been sliced and then layered on top of each other, or like laid out. I think you get like $250 if you donate your body to science like that. So, you need the money. All right, so this is a scanning electron microscope. It's called scanning because it sort of scans the surface. So again, if you were looking at the cells or a tumor or whatever, you would just take the tumor and then you would coat the outside of it, the outside of the cells, with lead or gold or some heavy metal. And these are the same cells, right, that I just showed you before. These are the ones that line your small intestines, and you can see the cilia, but can you see inside the cell? No, you can see really well on the outside of the cell, but you can't see inside. So in this instance, let's say that you wanted to look at you designed a cancer drug, and you want to see if it binds to the receptor on the surface of the cell. What kind of electron microscope would you use? Scanning. Now, you would also want to know if it gets taken inside the cell, right? So it's actually being delivered. So what kind of electron micrograph would you want to look at if you're looking at the inside of the cell? Transmission. Good. So you can see why you would need different microscopes for different techniques. This is more of a three-dimensional image. It's pretty easy to tell the two apart. apart. You just look at what it looks like. Question?
Was not sliced.
No.
That one said it aims to view the surface of the specimen. The other one is saying that it aims at microscopic sections.
So it goes through. It goes through the slices. So it's transmitting through as opposed to bouncing off the surface. All right. So that covers basically the kind of different kinds of microscopes. I mean, there are a few others that we didn't cover in class, but those are the most common ones.
So like in an atomic microscope, like when they look at the atom, do they just see the nucleus of the atom, or do they see the atom as a whole?
No, so you can't really penetrate the electron cloud. That violates quantum mechanics. So basically, you see it as like a space filling model, like the little balls.
I've seen like a video of iron under an atomic microscope, where they went down and it kind of looked all rough. And they got all the way down to like the atomic structure of the metal, but like it didn't look like it was like an atomic, like a cell, like almost like a big massive.
I haven't seen that image, so I can't really comment on it. But the ones that I have seen generally show the three-dimensional space filling models, or in some instances they'll show the protons, right, when they're ionic charges. So like hydrogen, you can see the nucleus because there's no electrons. But electrons are weird. When you start messing with electrons, you end up with these sort of quantum issues, which I don't really want to get into for this class, quantum mechanics. Anyway, all right, so self-fractionation, we did this in the lab. You guys did that in the other lab too, right? So self-fractionation is just what scientists do in order to study the components in a cell. What do we want to study? What are we interested in? Why are we doing this? Why do we do self-fractionation? What do we look at? We looked at organelles. Why do we do that? Just to look at them? Ooh. What? I mean, why would scientists do this?
See how they act
Understand.
understand how you work.
Well, yeah, so we want to understand their function. That's the reason for self-fractionation. So on the exam, if I ask you, what's the purpose of self-fractionation? What are you going to tell me? Right, we're not the cell, but organelle. What's going on with the tracking on this camera? It keeps going over there.
I think there was a line here.
Whatever. I don't think it's working.
You just stand over there.
Yeah, I'll just stand over there. So, anyway. Now it's over there. Nobody can move. It's very sensitive today. Go anyway. So, you guys know that whenever we did the centrifugation, we got two components, which were supernatant, which is the liquid on top of the other part, which is the pellet. So anytime we separate things on using a centrifuge, we're going to get two phases, the supernatant and the pellet. This camera's pissing me off, actually. So, any questions about that? So, how does a centrifuge separate things? Based on what? Mass, right? So, the things that are heavier, they come out first or last? Is it really? I don't think that that's what it's tracking. I think it's pretty possessed, actually. I might have to shut it off. Anyway, we'll see what happens. Alright. So just make sure you guys know the terms. Homogenate is when you buy your milk, right? It's homogenated. That means that they've mixed the fats and all the other stuff there we go. So that they all seem uniform. So homogenate is the result of us blending everything together. Yeah, you guys can't see this because I have the monitor, but it's the camera's jumping all over the place. So centrifugation, you know what that is, right? And we separate things based on the mass because we spin it and increases the force of gravity. Supernatant, decant, do you know what that means? It means to pour it off. So that's what you guys did. And then the pellet. Alright, so you guys should already know what prokaryotic cells are, but let's just go over it again because we're gonna look at the internal components of it. So do prokaryotes have organelles? No, so that'll be easy to study, right? Do they have a nucleus?
Yes.
What does prokaryote mean? It means before nucleus, right? So prokaryotes don't have a nucleus. What about cell walls? Do they have those? Yes. They also have cell membranes. What are those membranes made out of? No. No. That's the. No. What makes your cell membrane? What is the component of your cell membrane? Phospholipids. Excellent. And then ribosomes. You guys remember what those are?
Engineers.
They make the proteins. That's right. They read the message in the RNA and they make the proteins. Since prokaryotes don't have a nucleus, we just say that their DNA is kind of out there in this area called the nucleoid, which is a rare term, but you might want to know that. And then sometimes they have a capsule, and we'll talk about that later, but it's like a gelatinous coating that can go on the outside of the cell wall. And it helps them evade like organisms' immune systems. Mycobacterium, tuberculosis, and pneumonia make good capsids so that they evade the immune system. This is what kind of electron micrograph do you think? Why?
No, it's a section.
Yeah, so because you can see the inside of the cell, so you know it's even though it's written there too. All right, so eukaryotic cells, do they have organelles? Do they have a cell wall?
Some.
Some do. What are they? Plants and fungi. Good. So it has a true nucleus. What's in the nucleus? DNA, right? And then there's other organelles that we haven't talked about, like the Endoplasmic Reticulum, which is shown here in blue, the Golgi, which is shown here in this sort of a pink color. What are these? Do you guys know? What do they do? They make ATP. Excellent. Peroxisomes we're not going to talk much about. They break down hydrogen peroxide and things like that. Lysosome, we talked about that. What does that do? Right, so that does the digesting of the components in the cell. Centrosome, we'll talk about this in chapter 13 and chapter 12. Ribosomes, you guys know what those are. And here you saw the things that we call microvilli, which are also known as cilia, that are found in your intestines. And then some cells have a flagellum, like in humans. Sperm is a good example. Any questions about that? So I'm just showing you this so you can get an idea of how this is laid out. The nucleus is generally in the center. On the outside of that is the endoplasmic reticulum, and then right outside of that is the Golgi.
So is that like where the amino acids sit?
The amino acids, they're just out here. They get put together by the ribosomes.
So it'd be like they sit throughout the Golgi.
No, so there are ribosomes on the rough endoplasmic reticulum. That's why it's called the rough endoplasmic reticulum. There's also ribosomes just out here, right? They're called free ribosomes. We're going to talk about the difference between the two in a second. Any other questions? All right. So, you guys ever seen any giant cells wandering around, eating people, flesh-eating amoebas? Why not? The blob? Why not? Why don't they exist? Why aren't there any giant cells?
The cell is the smallest.
Yeah, but why can't they be big? What prevents that? Right. So the cytoplasm, the surface of the cell increases a small amount compared to the volume. Like when you blow up a balloon, the surface area doesn't increase as much as the volume, right? The volume exponentially increases. So that's a major problem, right? Because you can only get so much gas across the surface of the cell. So it can't be so big because the inside part of it wouldn't get the nutrients or anything that it needed, and it would eventually die. So that's why you don't see giant cells. That's why you only generally see cells that are smaller than you can see with a microscope. And then there's a limit to how small a cell can be too. Why is that? Right. You could put 30 people in a Volkswagen, but that doesn't mean that they can do anything, right, in there. They can't, work. So, you can make a small cell, but at some point, there's not enough room for the enzymes and the other things to actually do their jobs. So then it would stop functioning and the cell would die. So that's why there's an upper and a lower limit for the size of cells. And then those organelles are partitioned off by internal membranes. So what's your membrane made out of? Phospholipids, right? And what kind of phospholipids? What's the arrangement of phospholipids that make up a membrane? Right, it's a bilayer, so it's 2 layers. So all of these organelles, like the mitochondria, the nucleus, the endoplasmic reticulum, what do you think all of their membranes are made out of? Phospholipid bilayers. That's exactly right. And why do they need that? Why do we need membranes within a membrane?
Because they each have their own function. Yeah. To facilitate certain dumping.
Regulate traffic in and out of the cell.
Right, in and out of the cell, but that's just the cell membrane's job, right? Once you get traffic in the cell, why do you need a membrane around the lysosome?
To keep them inside.
Yeah, this pH is 5 and this is 7.2 to 7.6. So without that membrane, if that wasn't there, could you get a pH of 5 inside of that? No. Also, there's another issue, right? There's enzymes in here. There's one called a DNase. Do you know what it does? Want to guess? Destroys DNA. So, would you like these to have no membranes? Then the DNase would just go in there and say, "Hello, DNA, I'm here to destroy you." Would that be good for the cell? No, because that's your only instruction book. Without it, you wouldn't know how to make you. So that would be bad. So it needs to sequester certain enzymes, right? That has to keep them apart from other ones, because that you don't want just let any old riffraff in. And then the outside membrane has to be able to maintain homeostasis. So that's why we have these internal membranes. All right, so we're going to go through the organelles individually. We'll start with the nucleus. What's the function of the nucleus? It contains the DNA, right? The nucleus actually has two membranes. So you can see that here. It's an inner and an outer membrane. This is the inside, and this is the outside of the nucleus. It has little channels called pores, nuclear pores. These things are gated, just like a fancy community. So it only allows certain things in and out of it. The inside of it is what we call the nuclear lamina, and that's made out of keratin. Do you guys know what keratin is? What makes up your hair, your fingernails? Yeah, it's a type of protein. So its job is to coat the inside of the nucleus so it maintains that round shape. So basically, in every one of your cells is a little fingernail that allows the nucleus to maintain its shape. And then the outer membrane is shared with the endoplasmic reticulum. So I don't know if you can see this or not, but maybe you can later on when you look at this in your book. But the inner membrane is here. It's continuous with the pores, and then the outer membrane actually can jut off and form the outer membrane of the endoplasmic reticulum as well. So I might ask you, what does the outer membrane of the nucleus share its membrane with? Yeah. I might ask you, what's the purpose of the nuclear lamina? Right. What's the purpose of the nuclear pores? Yeah, so it regulates stuff that goes in and out. Regulates traffic.
Now, are there different pores? Do each of the pores have different functions that allow different things in and out?
That's a good question. So the pores on the nucleus are a little more robust, I guess. They allow more things in and out than, say, the membrane of the cell.
So like if a bacteria goes into the cell, it goes all the way to the nucleus?
So the nucleus wouldn't allow, the pores are not big enough for a bacteria to fit in. A virus, some virus.
Nucleus, right? Affects the cytoplasm, right?
Well, it depends. Like a retrovirus would go inside the nucleus and then integrate into your DNA.
That's like HIV.
Right. Some viruses can, some can't. And basically, the cell, it's not like the cell goes, hey, come on in, virus or bacteria, having a little party in here, just hang out with us. It doesn't want those things in the cell. They exploit, channels that other things would use. Right? It's kind of like sneaking your friend into the party that they're banned from. So, chromatin, we talked about chromosomes, right? You guys know what chromo means? Chromo means color. That's chrono. So chromo means color, and some means part. Remember, that's why chromosomes got their name from the methylene blue stain that you guys did in lab. And so chromosomes are actually long segments of something we call chromatin. So what's chromatin? Well, chromatin is actually DNA combined with proteins. And there's a reason for chromatin. So each one of your cells that you will look at under the microscope when we do the lab that I wrote, that lab 11. But you can't see the nucleus of your cells without a microscope, right? Nobody can see their cells with their naked eye. But you have over 7 feet of DNA inside each one of those cells. So how do you get it in there? Just wad it up, throw it over in there. Alright, so I'll tell, since nobody's going to guess, I'll tell you a little story. When I was a kid, we used to go take glass Coke bottles, the 32-ounce Coke bottles, and go return those to the store, like Safeway, to buy money. You know what we were buying? We were buying candy. We were buying kite string because we had kites and we wanted to see how high we could fly the kites. So we had, you know, almost a mile of string, over 5,000 feet of string that we, you know, spent all day flying this kite. And then towards the end of the day, it's getting dark and your parents are like, all right, you got to come in. So what do you got to do? You got to start winding the kite string around the spool. Well, there's no way, that takes forever. So then what do? You just pull it in, right? And then you just throw it in the garage, your mile of kite string. And then your dad comes home and what does he say? What the hell is this mess, right? It's not organized at all. So your cells are like the spools, right? If you would never be able to fit that DNA in if you just did like I did and threw it all in the garage, in the nucleus. So you have to wrap it up, right? It makes it organized. So the proteins actually look like wheels, and the DNA wraps around those. So those proteins are called histones, and the DNA plus the spools are called chromatin. And that's what allows your DNA to be organized and packaged inside of those cells. Otherwise, it would never fit. Does that make sense? All right. So every organism has a different, well, not all of them. I mean, there's there's lots of species, but there's only certain numbers of chromosomes. So almost, you know, we as humans have a different number of chromosomes than, say, chimpanzees or elephants or wheat or pineapples. So do you think that the number of chromosomes dictates your evolutionary superiority or your intellectual superiority?
Based on that.
Because if that's true, humans have 46 and chimpanzees have 48. Pineapples have more chromosomes than humans. Can you outthink a pineapple? I hope so. Not sure based on your test scores. Just kidding. You know, there's a Tool song. You guys know the band Tool? So it talks about the 46 plus 2, right? Which is, it's kind of like a satire, because what it's saying is that our next evolution would be to develop 2 extra chromosomes, right? As humans. But if we had 2 more chromosomes, what would we be? Chimpanzees. So it's kind of an oxymoron, right? It's not really an evolutionary advantage to have extra chromosomes, because in some cases it's a disadvantage. Elephants have 56, woolly mammoths have 58. So remember we talked about the Japanese, they want to take the DNA from the woolly mammoth and clone it into an elephant embryo and then implant it into an elephant surrogate, and then they think that that would give birth to a woolly mammoth. But there may be an issue, right? Because what's the most glaring issue that you can see? They have different numbers of chromosomes. So that could be a major problem. We just don't know. But this is most important because we're going to go over this over and over again. So all of you look normal to me, so I assume that you have 23 pairs of chromosomes, right? So that means you have 46 total. 23 you got from your mother, 23 you got from your father. Most organisms have 2 sets, right? Half they got from their mom, half they got from their dad. We call that diploid. We'll talk about that again when we get to chapter 14 in genetics.
Or chromosome, so is it
So there's in humans the dosage is really critical, and so if you don't have enough of something, it's almost always fatal. So there are no instances where you have individual with one, two, few chromosomes, except there's a condition called Turner syndrome, which is you only get one X chromosome. So that's survivable, but anything else with one, too few is death, right? You would never be born. You would be, a miscarriage. There are three chromosomes that allow for organisms to live, to be born, but most of those, they don't live past the age of 2. So there's Edwards syndrome. There are some, but they involve the sex chromosomes, like Klinefelter's. But the most common one you guys probably know about is the one extra number 21, which is Down syndrome, trisomy 21. And the reason and that's the most survivable. So all the other ones, generally, humans don't live past the age of 1 or 2. Most of them die before they're 6 months old. But the reason is because the chromosomes are numbered based on their length. So 1 is the longest, and then it goes to 22, which we have a pair of those, which supposedly is the shortest, but someone screwed up when they were actually looking at it. And it turns out when you sequence the DNA, you count the letters, 21 is actually the smallest. So the reason 21 is probably survivable is because it has the least amount of information. That's a good question.
What is that?
The what? Down syndrome is trisomy 21. You don't need to know that. We'll cover that in genetics in chapter 14. But that's when you have one extra number 21 chromosome. I can't turn it back on because it's blinking at me. When it stops, I can. I apologize. I've asked them to come in here and fix it, and apparently they, it's like one of those things where nobody's gonna sit. Yeah, it's not like it won't just break. So then no one sits in here for an hour and goes, "Oh yeah, I see what you're talking about." They just think I'm crazy. All right. So anyway,
The nucleolus is this area. So this is the cell, right? This is the nucleus near the purple part. And then look at cells. There's always a dark area that stains, and that's called the nucleolus.
and the reason that it's dark is because it is the site of the synthesis of the most important components that make up the cell. So what do you think the most important thing in the cell is? No? It's not RNA, and it's not DNA. Ribosomes. Ribosomes are the most critical thing. Without those, you can't make the proteins that you need to live. And so the most important part of the ribosome is that ribosomal RNA. Remember I told you it tells it how to read it? So, I was telling my other class, it would be like if I said, I need to get some food for my snake, so I want you to go get a rat, and you read it backwards. What would you bring me to feed my snake? Would my snake like that? No, it would kill my snake. And it would happen the same thing inside your body, right? So, I don't keep snakes anymore, but I used to. But anyway, so if the RNA got flipped over and it got read backwards, it could make something that would be harmful to you, right? It might hurt you if you ate tar. Or rats, I don't know. Some people eat rats, right?
But the city too, there's like I read an article like 2 weeks ago. This guy who survived 18 years in a mountain in a mine. He was trapped in an earthquake, and he survived off the rice that was in there, off rats inside the mine.
I'd eat a rat if I had to.
For
Wouldn't have a
17
problem with it.
years, the only thing that kept him alive was there was a vent where he was, and that was still circulating air. Found it by accident.
That's crazy. So, this is the site of the ribosomal RNA that's synthesized here. And remember that the ribosomal RNA plus the proteins make the actual ribosomes themselves. It's just a really big site of activity, right?
A lot of
Yeah, there's a lot of there's a lot. The DNA is always being expressed there. Those genes are always on. They're always being made into RNA. So you can literally see the production of ribosomal RNA coming out of there. All right. So speaking of ribosomes, you guys know what they are, right? They have two subunits, a large subunit and a small subunit, and the mRNA goes in between those two. The RNA, what kind of RNA makes up ribosomes? Yeah, it's ribosomal RNA. Yeah, rRNA. And then proteins. And these things, their job is, they're like the chefs of the cell, right? Whatever message they get from the mRNA, they cook it in the form of proteins, right? So your eye color, your hair components, your skin, everything that you're all proteins, right? DNA is just an instruction code. It's a blueprint, right? Is a blueprint ever going to build you a plane or a car? No, it's never going to make anything. So the ribosomes are what turns the instruction book into something physical that can be used, like enzymes or whatever. So they're really important. There are two kinds of ribosomes. There are free ribosomes and there are bound ribosomes. And they both have a different role. So let me see if I can kind of explain this. Yeah, I can turn that on. You guys just tell me when yours goes out, because I know it's going to happen soon. Yeah, if you guys have like laptops or tablets or whatever, it might be helpful to bring the slides in with me until I can prove that this is really happening and not just a figment of my
Why don't you
imagination.
let them watch the lecture video?
No, we told them to come at 10.
Yeah, can you guys show up at 9:30? They'll be like, yeah, whatever. Yeah.
This one goes on at 9:30 p.m.
Okay. So here's the nucleus. Here's the endoplasmic reticulum, and there could be ribosomes that are on this, or there can be ribosomes that are out here that are free, right? So those two dots represent the two separate subunits. So let's say that this thing is synthesizing a protein. And you guys, we haven't done the enzyme lab yet, but did you guys do it today? Okay, so the other lab, they did the enzyme lab, and we're going to do this on Thursday. So the guys that had lab today, what enzyme did you look at? Amylase, and what does it break down? And where's it found? In humans. In your saliva, right? So let's say that this is an amylase enzyme. And so this is what your salivary glands look like. It's not an exact representation, but it's pretty close. So here's your teeth and here's your gums. And there are cells that line this salivary gland, just like this, right? And so this job is to make saliva with the enzyme amylase in it. But it's made on the inside of the cell, right? So how do you get it outside of the cell? You could send this to the edge, right? But it's kind of like a soap bubble. So whenever this merges with this edge, it's going to create a bubble, phospholipid bubble around that. Its job is to digest the food that you eat. So will it function that way? It's surrounded by a phospholipid bilayer. So then you couldn't make it on free ribosomes. Well, let's look at the bound ones, right? So let's say that amylase was synthesized inside the endoplasmic reticulum. So the ribosomes would transfer inside this. Now, this comes to the edge, and what's going to happen to this when it gets to the edge of the phospholipid bilayer? Same thing that happened here, right? This will pinch off and form a bubble, right? And then it'll go to the outside of the cell, and when it goes to the outside of the cell, it will fuse and release it. So if you want something to be exported out of the cell and be functional, like insulin's made this way, right? It goes into your blood. It's not enclosed. Amylase, proteins that are found in mammary glands, right? Like milk. They have to be exported. So those have to be synthesized on the rough ER, which we consider the bound ribosomes, and not the free ribosomes. Because free ribosomes, they can only make functional proteins that are going to work inside the cell, inside the cytoplasm.
So how is the phospholipid layer generated?
Yeah, so that's a good question. So the smooth endoplasmic reticulum synthesizes the phospholipids, right? And also there's a shuttle. So this is a good question, because when this happens, right, you're taking part of the membrane from the endoplasmic reticulum and you're putting it into the external membrane of the cell, right? So what happens to the membrane of the cell now? It's going to get bigger, and this is going to get smaller. And remember, there's a limit, so that we can't just let this thing grow out of control. So there's shuttles that will bring the membrane back, what are called vesicles, and there's also the ability of the endoplasmic reticulum to synthesize phospholipids that it can replace into its membrane. Any questions about that? All right, so let me show you guys a video real quick about how this works, because it's kind of. Thank you. I don't know why this is so slow. Maybe we didn't pair it right up.
Cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. The plasma membrane. They leave the cell.
Okay, what's this right here? What's this? What's that? Golgi, what's this? Good.
Well, instead, by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus,
So you guys see how that works? And it can reverse it
This
too.
process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Okay, so we just talked about the vesicles, right? Those are the things that surround it. And then the endomembrane system is a system of organelles that either physically share membranes. So remember we talked about the nucleus and the endoplasmic reticulum sharing that outside membrane. So that would be part of the endomembrane system. But also, you saw in that video where the ER, it will move part of its membrane and that will fuse with the Golgi. So they also share membranes. They're not directly connected, but they share them through vesicles. So that those are considered part of the endomembrane system as well. So we'll talk about how this endomembrane system works together to transport molecules. And then another thing I want to point out before I let you guys go is that remember you're making mRNAs in here, right? And those are getting translated into proteins either by the rough ER or the free ER. So even if you made an mRNA and made it properly, if it didn't go to the right place, it wouldn't function. So there are signals on the messenger RNA that tell it where to go, and those signals could get messed up too. So not only you could have an intact piece of DNA, a gene that's fully functional, and if the signal is messed up, then it wouldn't work. So it would almost be like not having a functional gene. Yeah, everything goes back to the ER. Yep, and it will break it down. All right, so I'll see you guys on Thursday.
Is there a limit
Yeah, size-wise, yeah.
Why does it need to go through the Golgi if it already got
So the Golgi modifies things. We'll talk about that on Thursday, but you know, you talked about the bad cholesterol, right? The LDL. So that's a lipoprotein. So what does that mean? It's a protein with a lipid attached to it, right? And there's glycoproteins, like your blood type, A and B, that has a sugar attached to it in a certain place. And the Golgi is what does that attaching. So it modifies the original protein. That's the job of the Golgi. And the Golgi also directs where it's supposed to go in the cell.
So I'm not here Thursday, but I did the lab today, the Tuesday class.
Okay.
Instead. And I just signed some more signage. But is there anything else I need to do to get the credit for that?
No,
Okay.
just remind me in the end if we don't give you your points or whatever.
Okay, cool. And then he assigned them like homework or like they need to make like a chart out of the data. Are you doing that too, or is that so I'm good?
Yeah.
Just, okay. All right. Thank you.
So, so now like I got 133 on the test with the 143 or something like 95%.
A what?
143,
What did you say the percent is?
95.
I don't know. I mean, I didn't do
It's the
math.
math.
Okay.
So it's really-
Okay, I trust you. So, what does that mean?
Oh, you feel better
Do
now?
you know how?
Feel better
Yeah,
now?
no, but do you know how upset I was today? I like, cause like when I took the test, I was so confident in my answers. I was like, "There's no way I missed more than 3." Right? There was 3 in my head that I doubted myself in, and I was like, I knew the one with the kinetic energy. How much did it take? How many degrees Celsius did it take for it to digest that? I did my math wrong and I got 2. And I was like, something's off, but I can't fix it. So I just moved on because I don't want to waste my time on one question. And then with the number 7, with the species differs, and I was like, so I answered 7 right, 7 wrong, and I answered 8 right. I don't know how I did that.